Well, as we've done with all of the transmission genetic lectures, we try to end them by working some problems, some example problems to show you, okay, what would we do? How would we handle it? Okay. Now, you may remember that when we did the transmission problems early on, I said, anytime you're doing one of these transmission word problems, I tried to give you a way to think about breaking it down. Very simple way. I said there's always A times B, so a couple of parents, and they produce C. And some of that information, or all of it, was provided to you. So you might do a top down or a bottom up. Well, as far as working the types of linkage problems that we're going to work, particularly from the test and off out of the book and others, there's three simple things you need to know. Okay, you need to, every time you start a linkage problem, first off, you ask, is it linked or not? In other words, is there any indication from the actual wording, or do I need to look and see if there's a violation of Mendel's second principle or whatever? I need to figure out whether it's linked. Sometimes that information is given to you in the problem. Sometimes you actually have to work through it. Once you figure out it's linked, then you have to worry about the configuration, right? Whether it's in repulsed or coupled configuration, because that'll often play a role in the type of answer that you expect. And then finally, we'll almost always ask you about distance. How far apart? If they are linked, how far apart must they be? All right. So these three things. So just write them down right now. Anytime you start a problem that you think it is definitely some type of linkage problem, at least a 2-point mapping problem, always have those three things in mind. Okay, and try to answer them either from the wording, or you'll have to answer them yourself by doing the calculations. All right, so let's take a problem. All right, a student has two dominant traits here. Okay, let me get my little pointer going here. Two dominant traits depend on a single gene. There's cataracts, which he inherits from his mother, which is a dominant. Cataracts is dominant over lack of cataracts. Okay, so eye conditions where we get proteins in the eyes and eventually affects our vision. And then there's polydacty, which he inherited from his father. Polydacty is when you have extra fingers or extra parts of fingers, usually at one of the terminal knuckles or somewhere that sort of pulls out, and you actually have additional fingers parts that will come out. Polydacty, as it's known, okay, quite common for a number of different genetic diseases and traits, or for just some environmental irregularities during fetal development. It's fairly common and easily dealt with if you want to deal with it. But in this case, it's a dominant trait. Okay, so there are forms of it which are simple dominant. So we're talking about simple Mendelian traits, right? Now, if these traits are tightly linked, what are you going to get? Okay, so you've got the student. He's got two parents, A and B, and he's C, and you're asking about C. Okay, so we're going to ask our big questions down here. Is there evidence here of linkage, or do we need to test for it? Is there configuration information? Is there distance information? Well, it turns out that there's a lot of information here, and it's all in 3 words right here. These traits are very tightly linked. Linked tells me, yes, they're linked, right? They're on the same chromosome. So we've already answered that. They're clearly on the same chromosome somewhere, so linkage. All right, configuration, well, we don't know that yet, but one thing we do know from those 3 words right there is very tightly. What that's telling me is they're really, really close together, or there's something preventing them from having any type of crossover and recombination. So as far as distance, yeah, they're tightly linked. They're so close that we don't have to worry about there being a distance effect. Okay, so just from those 3 words, we've answered 2 of the major parts of this question. That only leaves one part. And in fact, this whole problem is based around a configuration question. All of this stuff up here and the answers that you'll provide are all based on knowing the configuration of this student, of this individual. What does his genotype look like? Okay, and what form is it in? Now that you have to deal with the fact that you've got linkage. So let's take a look. The questions that would, the rest of the questions that come up here are, what's the probability that this student will produce children who have either cataracts or polydacty, one or the other, both cataracts and polydacty, or have neither, so it'd be completely normal, not have any evidence of cataracts and never have polydacty, okay? And then of course we ask you to explain your answer. Give me some kind of explanation. Well, okay, this is a configuration problem, and I'm going to select this as the answer, that the children will either have cataracts or polydacty, okay? I'm going to argue that you're highly unlikely to get any children to have both, and just in the exact same logic that you'd use for saying they won't have both. They won't have the complete lack of it either. The same logic for B and C applies. Why? Just think about it. Mom looks something like this, right? Mom has cataracts, which is a dominant, and she's contributed this, right? And she's wild type or normal for polydacty. Okay, that's the way it would look there. Okay, all right. The dad has polydacty, but he in fact is wild type for cataracts. He doesn't have any evidence of cataracts. So these are what they're going to give to the kid, right? Okay, because it says he inherited the cataracts from his mother. So we know the mom gave this. I mean, she could be plus plus on the other one down here, but we know that the student is going to have cataracts and the student's going to have polydacty, right? Because he inherited that from his father. The only way he would get both of those is for dad to give him this chromosome and mom to give him this chromosome. So we would actually, if we looked at the student, we would say that he looks something like this, right? And if you want to put this in some terminology that you might be more familiar with, this would be big A, little b, little a, big B, right? If you want to think about the A's and B's being the dominants, but there's no reason to. You can use the C's and P's here. Okay, so he's got this configuration. What is that configuration? Well, you got a 50-50 chance, right? Is it coupled or repulsed? Well, it looks like to me that the dominant traits are not together on a homolog, but they're crisscrossed from each other, which is a repulsed configuration. So the answer is pretty straightforward, okay? He's in a repulsed configuration. If they're very tightly linked, if this is linked to that, there's no crossover going to occur in this middle right here, then you can't get a dominant cataract and a dominant polydacty. So there's almost no chance that he's going to produce children to have both, right? Because these are rare diseases. It's not like his spouse is going to have it, okay? So he's going to produce gametes that look like that or gametes that look like that. In other words, what we have going on here is complete linkage, right? This defines it as basically complete linkage. Yeah, you could get some crossover, but it's highly unlikely. Most of its probability, the kids are either going to get this gamete right here, which means they'll have cataracts, or they're going to get this gamete right here, which is going to make them polydacty. Okay, so classic problem. Is there evidence of linkage, yes or no? Is there evidence? What is the configuration if you're linked? And then what is the distance? Well, we got two of those answered. Yes, it's linked, and the distance is essentially zero. We're going to call it complete linkage for these purposes. So all we're doing is a configuration problem. All right. Okay, so let's try another one. So in this case, you've got some plants that are produced, and they're heterozygous for leaf color, and exhibits red leaves and normal seeds, and cross another plant exhibiting the complete recessive. So once again, we're in a simple dominant recessive system. Again, in laboratories and other places and other problems, you may have to deal with codominance and other things, but right now we're just trying to get the basics down, so we're going to look at it from a simple thing. All right, here's the progeny. Question. Do these data support the hypothesis that they're linked? In other words, you're asked this time to figure out, are they linked? Okay, are they linked or not? Right? It's not being told whether they're linked. You've got to figure it out. And then it says if they are linked, what is the percentage of recombination? In other words, what's the distance? Right? So you're asked for two parts. You aren't asked for configuration here, but you're asked for two parts. So let's take it and write down the given information. You've got one individual with red leaves and normal seeds, which we take to be the dominant, okay? And you've got another individual who has the reverse of these, which are green leaves and rounded, all right? That is a classic cross of a recessive back cross, right? I mean, it is just, we've got a dihybrid het. You can think of that as big A, big B, little b if you wanted, times little a, little b, little b. It doesn't matter in this case, but you've got dihybrid het crossed with, because it says it's heterozygous, right? Crossed with this, okay? So if we take, of course, the plus to be wild type, if we assume that the red is wild type and the normal is wild type, we're making this cross, okay? Mendel's second principle says we should be able to separate the color of the leaves from the seed shape. We should be able to produce the segregation ratios, which are 1-to-1 for both of those. And then we should be able to put them back together to figure out what our expected genotypic and phenotypic ratios could be. Now these are assortment ratios. Well, obviously they're going to be the same one we're doing for all of these linkage problems, right? They're 1 to 1 to 1 to 1. That's just exactly the cross we make, no matter what. Okay, unless we've got a sex-linked or a lethal or something, but for these it'll work. Okay, so can we answer those original questions? The original questions were, based on these data, is there evidence for linkage? Well, what is evidence for linkage? Evidence for linkage is a violation of Mendel's second principle, right? Mendel's second principle says I have about 1/4 be red normal, 1/4 be red round, 1/4 this, 1/4 this. So 1/4 all the way down, okay? If I do that, I add all these up, I'm going to estimate my expected to be, of course, the same all the way down, 125, 125, 120 minus 125 squared divided by 125. Add all those up, I get a chi-squared value of 2.2. All right, that's well below the table value, so I have no reason to reject the idea that these are not following Mendel's basic ideas. So do these data support the idea that these are linked? The answer is no. Why? Because we find Mendelian independent assortment. We find evidence for that. Now they could be linked, okay? But it's no real evidence for that based on this. If I were to answer this question the best way I could, the data do not support the idea that they're linked, okay? And why not? Because I did a chi-squared test using the Mendelian assortment expectations, and I found that it fits within the boundaries of a chi-squared, okay? Now the next question is a little odd, and it's there to make us think about something. If they were linked, okay, what is the percentage of recombination? Well, they're not linked, so I don't have to answer it. No, you do have to answer it, okay? In this case, why? Because if they are linked, what must be going on? So it's saying, I don't care what your data say, I know they're linked. What must be the percentage recombination or the distance? Well, that's just to remind you that when you're doing 2-point mapping and you get above 50 map units, right? It's going to look like independent assortment. So these could be on the same chromosome, just in the same way that Mendel had some of his traits actually on the same chromosome as P's, but they're so far away, they're greater than 50 map units away, greater than 50% recombination, that it looks like independent assortment. Almost every time they go through it, you would see crossover and recombination somewhere in between anyway, so it looks like independent assortment. Okay. All right, so that's a good, nice one to work through. All right, so let's try one more. Let's take some mutant alleles. Now we've got sex-linked ones, right? Now, fortunately, it's in Drosophila, and we're going to try to look at them. These are got 2 recessives, so we've got cross veinlessness, which is in the wings, means they don't have these little cross veins, okay? And singed bristles, which are these little bristles that have this mutation where they just curl up and they look like somebody kind of like the fly, went too close to a fire for a moment. Okay, all right. Now, this case, we're not going to cross this anybody. We're just going to look at the outcomes from a heterozygous female. I told you back earlier that earlier lecture that sometimes you don't even need to do the cross. You can just look at the eggs, right? And this is particularly true here. We can just look at the eggs of this X-linked locus, and we can figure out what's going on. So here's what we get. We looked at the eggs. We get some that show up being both cross-veinless and singed. We get a comparable number that are both wild types, so they're normal winged and normal bristles. We get a few that turned out just to be cross-veinless, but normal as far as bristles. And then we get just the complement of that. We get a few more that look like they're going to be, just singed, right? All right, now remember the 3 questions. Is it linked? What's the configuration? What's the distance? Well, it says they're linked right here, right? So we don't even have to answer that. It says 2 X-linked loci. So they're both, we know they're linked. So obviously the questions are going to come up about either a distance or configuration or both. And sure enough, first question is this individual, is she in a coupled or repulsed configuration? Okay. Remember what we look for. We look for, in this case, we got simple dominant recessives. So if we've got 2 dominants together and 2 recessives together separately, then we've got coupled. If we've got a dominant and a recessive and a dominant, then we have repulsed. Well, obviously the largest number is what you look for. That's the ones that are parental-like. The least likely thing you expect to happen is crossover. So the most common thing you expect to see is not crossover, right? Is parental-like. So always pick the biggest pair to be the parental-like, and they've conveniently put them together for you here. And you can see that the recessives are together and the dominants are together, right? So this must be coupled. Very simple answer, okay? All right, we've answered the first of those. Okay, so we know they're linked, A, we've told that. We figured out pretty quickly that they're coupled by looking at the configuration that you see in the outcome. And then you got to know the distance. How do we calculate the distance? We use the recombinants. Well, these are the two recombinant pairs, right? So we're going to add those up, divide by the total, and bring it up into percentage. So we've got what? 56 plus 49 divided by, looks like apparently that adds up to 5,000. It's about 0.021, which if you bring it up into percentage is about 2.1%. So that's about 2.1 map units. So they're linked and they're really tightly linked together. They're very close, but there is enough to have crossover and produce a new progeny. One of the reasons we use fruit flies, besides not having that many chromosomes to deal with, like humans, which take forever to do this mapping, they only have 4 pairs, right? You can get lots of progeny, and even things that are really, really close together, which you wouldn't get many progeny, you still can get enough that you can actually come up with some pretty decent estimates of distance, right? And then there's a part C, which kind of relates to what you'll be doing when you try to put these together in 3-point mapping and other places. If one of the loci, okay, C, is known as a fixed distance, okay, that's how you would work this, is a fixed distance. It's 10.9 map units from the end of the chromosome, and we figure out that S locus is actually closer to the centromere. So we've got the C locus down here somewhere from the top, and if my knuckle is the centromere, we know that the S locus is down further, it's closer to the centromere. So what's the exact distance that the S, well, it turns out to be just an additive function, right? So if we know that this one of them is 10.9 map units away from the end, okay? And we know that the map distance between that one, the C and the S, is around 2.1. We can simply add those together, and it comes out to be about nice 13. So we now know that this one's the first locus, C locus, is about 10.9 units down. And then a little further down at 13 map units from the end is the other locus, which is the S locus for singed bristles. Okay. Well, that's the basis for how to work these problems. People struggle with them until they just go work a few. I mean, once you've done a few, you'll just go, oh yeah, and once you use this sort of idea that you need, just kind of like, pulling out Mendel's Big Six and holding it up. If you think about the fact that I'm going to be asked, are they linked? What's the configuration and what's the distance? That's the things that get put in there, okay? And you're provided some of that information, just like back when you did A times B gives me C, you might get A and you might get B, or you might get A and C, whatever. You could figure out the rest of it, right? Same thing here. So those are basic problems, all right? And that pretty much ends the whole discussion that we're going to have about transmission genetics. And in fact, linkage has sort of moved us down the hallway to the next big area that we're going to talk about, a new area for us. We've started covering sort of molecular genetics, and we did a little of that. We'll pick some more up in a little while. We've now gone through transmission genetics, okay, big area of genetics, big room in this sort of layout. And now we're going into a whole new room, okay, a whole new area, which is chromosomes, cytology, the study of chromosomes themselves. So this has kind of moved us toward that idea because we talked about things that are linked on chromosomes. But the next few lectures will be about chromosomes and chromosome variation.